everyone, welcome to my channel. In this video, we are going to see general physiology topic, that is transport across the cell membrane. Now, this transport is divided into two parts, two divisions, passive, active. Passive does not require energy and active requires energy. Passive is further divided as simple diffusion, facilitated diffusion, non-ionic diffusion and osmosis, whereas active is primary and secondary. Let's see the passive first. So we will move on to simple diffusion. Now, simple diffusion occurs in the substances that can readily cross the cell membrane. So these will be your gases, and as cell membrane is made up of phospholipids, the lipid soluble substances like alcohol, steroids can pass through it. Now the principle for the simple diffusion is Fick's law of diffusion, where J is equal to minus dA delta C upon delta X. J is the net transport, D is diffusion coefficient, A is surface area, delta C is concentration gradient, and delta X is thickness of membrane. And if you see this minus sign here, this minus sign has no mathematical significance. It just shows that the diffusion is happening from high to low concentration. Now this D here, that's diffusion coefficient, this is directly proportional to lipid solubility, inversely proportional to molecular diameter, and inversely proportional to the square root of molecular weight. So if you want to decide the substance that will diffuse faster, you will first compare the lipid solubility, then the molecular diameter, and then the molecular weight. So what are the factors affecting the net transport? One is the surface area, another is concentration gradient, then there is thickness of membrane, and these are the three lipid solubility, molecular diameter, and molecular weight factors affecting D, that is diffusion coefficient, ultimately affecting the net transport. Let's see the clinical aspects of these factors affecting. Like, you know that surface area is directly proportional to the net transport. So in emphysema, there is the breakdown of alveolar walls, or the surface area of the alveolar walls decrease for the exchange of the gases. Therefore, the diffusing capacity decreases. Let's see about the thickness of membrane. Now, you know that the net transport is inversely proportional to the thickness of membrane. In pulmonary fibrosis or pulmonary edema, the thickness increases and the diffusing capacity decreases. Moving on to the another division of passive transport, facilitated diffusion. Now, what's the difference between simple diffusion and facilitated diffusion? Both does not require energy, but facilitated diffusion is facilitated by carrier proteins. Now, carrier proteins, you know, we have studied it in 11th, 12th, that there are many properties of carrier proteins. The two which we are going to discuss here are specificity and saturation of carrier proteins. Now, carrier proteins are specific for substance or similar structure substances. Like, for example, GLUT1 is a carrier protein which is primarily for glucose, but as galactose is structurally similar to glucose, galactose can also be transported by GLUT1. So this will be an example of competitive inhibition, because when galactose amount is more, then glucose absorption is less. Let's see the saturation. Now here is a graph plotted where there is rate of transport versus concentration gradient. Now the black one is simple diffusion and the orange one is facilitated diffusion. Let's see the simple diffusion first. We know the net transport is directly proportional to the concentration gradient. So as the concentration gradient increases, the transport or diffusion increases. So simple diffusion will be a straight line. That's it. Coming to the facilitated diffusion. Initially, facilitated diffusion rate will be faster than that of the simple diffusion. This is because it is facilitated by a carrier protein. But just when this rate reaches Tm, that point carrier proteins get saturated, and hence the rate does not increase after that Tm point. So this orange one is your facilitated diffusion graph based on the saturation property of carrier proteins. Example of facilitated diffusion: glucose transport, then amino acid transporters and urea transporters. Urea transporters present in nephrons are UTA and present in RBCs and vasa recta are UTB. Again UTA are 1, 2, 3, 4. Moving on to the next division of passive transport, that is non-ionic diffusion. Now let's see a scenario in collecting duct cells. In collecting duct, the glutamine which is formed there is converted to ammonia by the enzyme glutaminase. Now this ammonia formed in the collecting duct cells will be more as compared to the ammonia present in the lumen. So there's this concentration gradient which pushes the ammonia into the lumen. Now in the lumen, towards these collecting duct cells, there are these H plus ions available. As soon as the ammonia diffuses into the lumen, these H plus ions combine with ammonia and form ammonium ions and traps these ammonium ions into the lumen. So what's the difference between a simple diffusion and non-ionic diffusion? If the ammonia would have been diffused and remained as ammonia in the lumen, then we could have called it a simple diffusion. But here, the ammonia is diffusing and it is ionizing to the NH4 plus or ammonium ion after diffusion into the lumen. Now, secretion of ammonia, it occurs at two places, PCT and collecting duct. But the mode of secretion or the mode of transport is different. In PCT, there is simple diffusion, whereas in collecting duct, there is non-ionic diffusion. What are the examples of non-ionic diffusion? Excretion of weak acid or bases by kidney, absorption of weak acids, mainly salicylates, by stomach, and absorption of bile acids by distal ileum. Next is osmosis. Osmosis is simple diffusion but more specific to water. So water from diluted solution moves to concentrated solution. These are the four divisions of passive transport. Moving on to the active transport. Active is primary, secondary. Primary has sodium potassium ATPase pump, hydrogen potassium ATPase proton pump, which is present in parietal cells of stomach and collecting duct, and SERCA, that is smooth endoplasmic reticulum calcium pump. The secondary transport, secondary active transport, again two types, counter transport and co-transport. This secondary active transport is dependent on the activity of sodium potassium ATPase pump, which is actually primary active transport pump. Let's see the sodium potassium ATPase pump in a bit detail. Now sodium potassium ATPase pump is ubiquitous, that is, it is present in all cells. Net loss of one positive charge occurs through sodium potassium ATPase pump. What is the actual thing happening? 3 sodium ions move out of the cells and 2 potassium ions move into the cell. So there is net loss of one sodium ion or one positive charge. So it is an electrogenic pump and contributes to RMP. You can see here, if the RMP is minus 90 millivolt, normally if you see the nerves and muscles, then contribution of Na plus and K plus pump is minus 4 millivolts. Now there's something called as coupling ratio. How can you say that it is 3:2 or 2:3? We know that 3 sodium ions move out and 2 potassium ions move in. But how can you say, if you have a question and you get 3:2 and 2:3, which one will you say more correct? Then the answer is 3:2, that is sodium is to potassium. The reason here is the movement of sodium and potassium efflux and influx is in two-stage process. The sodium efflux precedes potassium influx, and hence the coupling ratio accepted will be 3:2. Now we have seen that the contribution of Na plus K plus pump to RMP is very less, -4 millivolts. So what is the main function of sodium potassium pump? It is the cell volume regulation, because it causes net loss of one osmotically active positive ion, that is sodium ion. Now sodium potassium pump utilizes 1/4 of the total energy generated by cell, but in neurons it uses 3/4 of the total energy generated. And this sodium potassium pump is important contributor to basal metabolic rate. Let's see the structure of the pump. It is a heterodimer with alpha and beta subunits, and alpha being a larger subunit. Now let's say this is a cell membrane, the ECF, ICF, alpha, beta units. Now here in the alpha unit there are 5 sides. On the ECF side there is K plus binding site, UABN binding site, then sodium binding site, on ATP binding site, phosphorylation site on the ICF side. So ECF has 2, ICF has 3. So K plus from outside will bind to this site and then enter inside, whereas Na plus from inside will bind to this site and go out. There's something called as UABN binding site. UABN is actually a plant glycoside, and digitalis is derived from UABN. And this digitalis binds to the UABN binding site. What is this digitalis? I know you have not started doing this, you don't know much at the start, but digitalis is a drug that strengthens the contractions of the heart muscle. So this digitalis which we give, or the drug prescribed by the doctor, is binding at UABN binding site towards the ECF on alpha subunit of the Na plus K plus pump. Then of course there is ATP binding site and phosphorylation site on ECF side on the alpha subunit. If you see the beta subunit, then there are 3 extracellular glycosylation sites. That is, there are 3 glycosylation sites on the ECF side. Now let's see the hormones affecting sodium potassium pump. Now how will the hormones affect? Either by increasing or decreasing synthesis or by affecting its activity. Let's say thyroid. Thyroid increases the synthesis of Na plus K plus pump and does not affect its activity. Aldosterone increases synthesis as well as activity of the pump. Insulin increases the activity, does not affect the synthesis. Dopamine decreases the activity and does not affect the synthesis. And ANP, that is atrial natriuretic peptide, this decreases the activity of Na plus K plus pump and does not affect the synthesis. Now remember one thing which will help you ahead. Peptide hormones cannot affect the number, whereas the steroid hormones can affect the number. Why is this so? Because steroid hormones can enter into the cell and affect the nuclear activity. You have studied it in your human physiology, basics at least, in your 11th standard, and that tells you that steroid hormones can enter the cell and affect the nuclear activity. So any synthesis related changes will be caused by steroid hormones, whereas peptide hormones will only affect the activity and not affect the synthesis. But here's the special case of thyroid hormone. If you see, it is a peptide hormone, but it behaves like a steroid, and so it is affecting the synthesis. There's this application of diabetes ketoacidosis. There's a patient of diabetes ketoacidosis and he is in a comatose situation. What is diabetes ketoacidosis? A diabetes patient producing excess of ketone bodies, and as ketone bodies are acidic, we are calling it as ketoacidosis. Now the treatment for these patients is IV insulin. Now what will happen because of this IV insulin? Insulin increases the activity of sodium-potassium pump, we have seen here. So as this increases the activity of sodium-potassium pump, the movement of K+ into the cells will increase, and this will cause significant hypokalemia. And this is the common complication seen in diabetic ketoacidosis patient. So to prevent it, potassium is given. Moving on to secondary active transport, it is dependent on the activity of Na+K+ pump, or the primary active transport. Now how is this happening actually? Let's say this is my PCT cell. This one is my PCT cell. And this is the blood vessel near the cell. Now there are two sides of this cell. One is the luminal side. You can see here lumen. So this is the luminal side. And this side is basolateral side. The basolateral membrane is here near the blood vessel. Now always remember, sodium-potassium pump is present on the basolateral membrane. So you can see here sodium-potassium pump, and it is pumping 3 sodium outside and 2 potassium inside. So there is net loss of 1 sodium ion, and we say there is sodium ion concentration decrease in the PCT cells. So this primary active transport pump, that is sodium potassium ATPase pump, creates gradient for sodium. Now as the sodium is less, on the luminal side there are the pumps from which the sodium will enter into the cell, and the energy released by the entry of the sodium is used by an other ion to enter or exit the cell. This is your secondary active transport. So if the ion is entering together with the sodium, then it is co-transport or sym-transport, basically in the same direction. If the ion is going in the opposite direction, that of the entry of sodium, that is entering out or exiting out, then that is counter-transport or antiport. If there is no primary transport, then there will be no secondary transport. And the inward movement of sodium in secondary transport is leading to the active transport of other substances. Let's see some examples of co-transport and counter-transport. Co-transport, in PCT there is sodium, glucose, amino acid and inorganic phosphate pumps. In thick ascending limb, it is sodium, potassium 2 chloride pump. DCT, there is sodium chloride pump. In intestinal epithelial cells, there is sodium, glucose and amino acid pump. In distal ileum, there is sodium bile acid pump. And in thyroid cells, there is NIS pump or sodium iodide symport. In counter transport, we can see PCT, there is sodium hydrogen pump. In myocardial cells, there is sodium calcium pump. Now if you see here, we have discussed digitalis earlier, and we know that digitalis binds to ouabain binding site, which is on the ECF side of the sodium potassium ATPase pump on the alpha subunit. So here we are taking this digitalis, it is binding and inhibiting the sodium potassium pump. So what will happen if the sodium potassium pump gets inhibited? the Na plus, which was usually decreasing because in the cell, because of the primary active transport, will not decrease. So Na plus is adequate. So if you see on the luminal side, the Na plus which was coming inside will stop, and hence the calcium which was going out with the help of the entry of Na plus in this secondary active transport pump, this Ca2 plus will not go out, and there will be increase in calcium intracellular concentration. This will lead to increased force of contraction, that is, it is positively inotropic. Moving ahead to a completely different type of transport, that is exocytosis. What happens in exocytosis? Let's say this is your cell and there are some units like some particles. Let's say this is a body which needs to be engulfed inside, or we are seeing exocytosis. So this is a body inside and this is the body which needs to be given out or exocytosed. So what will happen? This cell has this body has, let's say, a cell membrane around it or a membrane around it of this color. Okay. Now, when it's going to exocytose, this will be this way. So here comes the addition of this membrane here, and the substance is thrown out. So what you can see is the membrane that was here, this red color membrane, is now added into your cell membrane or plasma membrane. So you can see the total area of the cell membrane increases because vesicular membrane becomes part of the cell membrane. What are the requirements of exocytosis? Calcium, ATP, and there are docking proteins. What are these docking proteins? These proteins help the vesicles to attach the cell membrane. Syntaxin or T-snare proteins, synaptobrevin or V-snare proteins, and SNAP25 are examples of docking proteins. Now you will study ahead that Botox or botulinum toxin causes flaccid paralysis. How does it causes flaccid paralysis? This is because this Botox inactivate the docking proteins. And you know that there is exocytosis of the acetylcholine at the neuromuscular junction. We have studied it in nerve physiology, or you will study it in nerve physiology further. Now, if there is inactivation of docking proteins, will there be the exocytosis? No. So, will there be the release of ACH? No. And as there is no release of ACH, that relates to the flaccid paralysis. You will still study it when you will be doing the neuromuscular junction. And this another toxin, that's tetanus toxin, which causes spastic paralysis, we will also study, or you will study it later. Now there are two pathways of exocytosis. One is non-constitutive or regulated pathway and another is constitutive pathway. What's the difference? Now you know that there is synthesis of enzyme, hormone and neurotransmitter, and there's a release. That's the normal thing about the exocytosis. But in regulated pathway, there is processing and storage. Whereas in constitutive pathway, there is no processing and storage. Example of non-constitutive or regulated pathway is secretion of insulin by beta cells of pancreas. There is pre-proinsulin, then proinsulin, and then insulin plus C-peptide. Similarly for glucagon by alpha cells of pancreas, there is proglucagon and then glucagon. Then there is acetylcholine at NMJ or neuromuscular junction. Secretion of enzymes by pancreatic acinar cells. Secretion of serotonin by Raphe-Magnus cells. In constitutive pathway of exocytosis, the examples will be secretion of immunoglobulins by plasma cells. secretion of collagen by fibroblast. Then we move on to the endocytosis. Now what happens in endocytosis? Now let's say this is your cell and you want to engulf something from here. And it has to be engulfed inside or endocytosed. So how will you do that? So there will be, and there's yes, of course, there's a membrane around it. There's no membrane around it. Sorry, there's no membrane around it, and it's just open. So what will happen? This cell membrane will just go and form a loop around it like this. Okay. And now what will happen? This has to be broken down. So there's this broken down part. Let's me draw it in different colors so that it will be better for your understanding. Like I'll draw it with red color. And here if you see, this is how I've drawn, and this will lead to the formation of the vesicle inside the cell, which is endocytosed right now. So if you see, there is decrease in the total area of cell membrane. Now based on the substance, we say it phago or pinocytosis. If it is solid, then phagocytosis. Liquid, then pinocytosis. Requirements of the endocytosis: there is calcium, ATP and proteins, clathrin, caveolin, dynamine and pinches, cubulin, megalin. Now what is clathrin? Clathrin is required for receptor-mediated endocytosis. Caveolin is present in endothelial cells and it is required for absorption of nutrients. And dynamine or pinches, it pinches the vesicles from the membrane. So this thing happening here, this pinching off of the vesicle here is happening by your dynamine or pinches. Then cubulin and megalin are present in kidney and intestine. And this is the last transport mechanism, that is transcytosis or cytopempis. This occurs in the intestinal epithelial cells of newborn. Now you know the newborns or the infants up to 6 months of their life, they are having the breast milk. Now the initial breast milk, you know, it is called as the colostrum, and the antibodies present in excess or the colostrum is rich in IgA antibodies. Now these IgA antibodies are secreted in the lumen, is present in the lumen after drinking from the mother. The newborn has this IgA antibodies in the lumen. Now by endocytosis, these IgA antibodies are taken into the intestinal epithelial cells, and then by exocytosis, these IgA antibodies are passed into the blood vessels and then into the circulation. So this is the method by which the newborn is provided with the passive immunity. So if you see in transcytosis or cytopempis, there is both endo and exocytosis occurring. In adults, the food allergies occurs due to transcytosis of allergens. That's it for the transport of cell membrane, transport across the cell membrane. Please like and share this video and subscribe to my channel. Thank you.